Hi, I'm Camila Hoyos. I come from Colombia, and this summer I was working in Lee Labs. They were basically with non-seed plants, and I was working assembling 3 nuclear genomes. Why is this important? Because genomic data is critical for uncovering evolutionary relationships, as well as basically the genetics behind traits of interest. And there are like more than 4,000 genomes assembled for more than 1,000 species, but they're not for non-seed plants, and they basically work with hornworts and algae. I was assembling the genomes for these 3 species, which are algae species. How did I do that? So first, I did the pre-processing, trimming, and filtering. I removed the adapters using FastP, and I filtered the reads based on 2 scores, basically the length and the Fred score, aiming to conserve a good depth, a sequencing depth. Then I assembled the genome to a contig level using HiFi-SM, and I compared the results with the one that my mentor did using another program that's called Fly. Then that contig assembly I polished because there are repetitive regions that are hard to assemble with long reads using Medaka and short reads using PyPolka. And finally, I did the scaffold assembly using high-C sequencing data that basically tells us how the DNA assembles in a 3D way. Once I had the genome assembled, I annotated it, which is basically doing gene predictions. So first I identified the transposable elements using EDTA. Then I used RNA reads to detect exons and introns, and the output files of both I used to do the final gene predictions using Breaker. So as a result, we obtained 2 scaffolded nuclear genomes. The stats went out great. We have high scores for M50, high scores for BUSCO, which assesses like the completeness of the genome assembled. Here, there are the stats of how many genes were predicted, and these are kinds of transposable elements that were predicted as well. These are contact genome maps, which are like based off the high-C sequencing data, tells us how the DNA arranges in a 3D way, which like parts of DNA are close to each other. And this aids us to uncover how many chromosomes are in this species and what's the size of the chromosomes there. So for Gormanilla, we uncovered 17 chromosomes of between 5 and 12 megabytes. And for Stigoclonium, we didn't know it had 16 chromosomes between 7 and 15 megabytes. We're also going to assemble the nuclear genome for Odogonium. And although we did the DNA extraction and we got like a good quantity of DNA, we found out that the culture was contaminated, and that's why we ended up assembling the chloroplast genome and annotating it. Here's the map. And so, as a conclusion, we obtained 2 scaffolded genomes assembly for Gormanilla and Stigoclonium, and an assembled and annotated genome for the chloroplast of Odogonium. We need to grow the culture again for Odogonium, sequence it, assemble the nuclear genome, and annotate it, given that it was contaminated. But the results were great, and I learned a lot. Thank you.
So for your original assembly up there, where did the genomes come from?
The genomes? Okay, so this is interesting. Gormanilla was actually a species that our lab discovered, and they named it after Amanda Gorman. So we isolated them, and the Oregonium we bought. Yeah, they come from there.